Okay. All right, well, I've already shown you my artwork, which isn't actually mine, but I did want to make a comment that I've really enjoyed seeing some of the artwork in these presentations. I've become very interested in transdisciplinary thinking, and I hear people wondering, well, how do we make the holobiont concept more palatable or approachable? And I think artwork and humanities and other ways of thinking is going to offer us a lot more headway than thinking within the sciences. So collaborating with those people, I think, is going to be really promising. So with that, one slide, yeah. All right, so this is a piece of artwork I really enjoy because as an ecologist, this is how I view the world. When animals and plants first evolved, we heard this a little bit in the introductory lecture, that the world was already dominated by microbes. And so as these organisms evolved and their physiological processes are evolving and developing, they're doing this in a world surrounded by microbes. And so a lot of these processes are probably reliant or evolved under the context of these organisms. But unfortunately, I think this is the world we've created. Through pollution, anthropogenic change, climate scenarios, we're really altering a lot of the environment for both the macroorganisms that we think about, but also all of these microorganisms. And so my own group, as well as others, have sort of made a call that we need to consider these host-microbe interactions in aspects of conservation, wildlife management. But I'm going to make a call that we really need to partner with these conservation agencies, not just work within our scientific institutions in order to actually make progress. All right, so today I'm going to tell you two stories. The first one is longer, talking about host microbe interactions and amphibian conservation. I'm not going to move. And then my second story will be about conserving host microbe interactions in imperiled species under human care, specifically thinking about whooping cranes in the US. But we'll start with this first story, thinking about amphibian conservation. And when I first started the lab, this was work really started by now Dr. Samantha Fontaine, who's an NSF postdoc. And she was really interested in whether microbes contribute to thermal performance in ectotherms, specifically vertebrate ectotherms. It had already been shown in invertebrates, but we wanted to think about it in vertebrate systems. So to do this, Sam went out and collected a bunch of pond water. She put this pond water immediately on ice, drove it back to the University of Pittsburgh. At the same time and the same place, she also collected adult frogs, which we brought back to the lab, injected with reproductive hormones, and generated small little egg masses in the lab. And from there, we raised these tadpoles under different treatments. So we have some that we term colonized, which I think we can, as a field, work on a better term. I'm not sure what that is, so I think that's an opportunity for discussion. And then depleted, where we autoclaved the water in order to remove a majority of the microbiome, although I should note that this is not a germ-free treatment. There are still microbes in there, probably from our water system, but we call this a depleted treatment. I'm not going to actually show the microbiome sequencing showing that these manipulations are effective. You can find that in the publication. So we raised tadpoles under these different microbial environments, and then we performed a series of dissections in order to look at thermal tolerance and also just morphometrics. And so one thing we found is that when tadpoles were raised under this depleted water is that they grew significantly larger. So you can see this here in our colonized and depleted treatments with these depleted animals growing larger, which you can also see in a photo here. But we were mostly interested in thermal tolerance. And so in order to measure this, we conducted CT max trials, or the critical thermal maximum that these animals can withstand. And so we place tadpoles in individual beakers in this water bath. We then warm up that water at a rate of 0.5 degrees per minute. You're constantly watching the tadpoles and giving them very gentle nudges with a glass rod in order to make sure that they're okay. They'll be swimming around for a while, but eventually they're going to meet this temperature where they are unable to right themselves. This is not lethal, although it is in a few minutes. But at the time, they have to be able to recover from this in order to be able to measure this CT max. So if they die, we don't end up recording that measurement. But after this trial, we did collect microbiome samples. So what did we find? So here I have our graph of CT max, same treatments. We're able to push the colonized animals up to about 40 degrees. But when we look at our depleted animals, these animals fall out of the trial significantly earlier. We've repeated these results in another set of animals, and we've also shown it at different acclimation temperatures. So this is a rather repeatable effect. But you might be looking at the y-axis and thinking, okay, 0.4 degrees, like what is the ecological relevance of that? And we were faced with that very same question. So we actually had some of that CT max data from an earlier experiment, but these were actually the repeat experiments. So in the context of this experiment, we then had animals enter a heat treatment. So we had some animals that were retained at the 22 degrees treatment, and then some sets of animals were ramped up to 32 degrees. We then continued to ramp up this temperature up to 34 degrees for a period of 10 days, and then up to 36. We monitored daily survival, and this temperature regime is really meant to mimic heat waves, or these prolonged exposures to heat, which are predicted to become more common under patterns of global climate change, including in Pennsylvania. So I'll present the results in a Kaplan-Meier survival curve. So here we have days of the experiment on the x-axis and the proportion of animals surviving on the y. And so in the beginning portion of the trial, we did observe a significant difference between our groups, such that the depleted animals were dying significantly faster, and so faced about a 5 times higher mortality risk under this temperature treatment. When we pushed these animals to higher levels, this difference continued in the 34-degree section of the experiment. So statistics run with only within these days were still significant. But when ramped up to 36 degrees, this seems to be too stressful of a temperature for everyone. So we see complete mortality in both groups. But at least within these middle temperatures, we see these significant differences, such that the depleted animals face higher mortality in face of these heat stressors. So we then have been following up with some of the molecular mechanisms underlying these differences. So back to this image, I'm going to show that in the beginning of this experiment, we also conducted acute heat exposures and dissected animals after just 24 hours. And this is before we observed any mortality. Again, we knew a lot of this from previous experiments, how we were going to run this one. So we collected these animals at 24 hours and did a lot of different molecular techniques. So we have microbial metagenomics and also intestinal transcriptomic data. And are running a number of comparisons to understand colonization effects, temperature effects, and the interactions. And so for today, I'm just going to focus on the intestinal transcriptome. So this is how the host is responding to heat. And I'm really going to focus on the heat differences. So how do colonized animals respond to heat, and how do depleted animals respond to heat? And what we saw is that they actually have very different responses. So these Venn diagrams show the number of genes that are responding to heat in either the colonized or the depleted animals. So we see that our colonized animals have a much smaller transcriptomic response, whereas our depleted animals are upregulating and downregulating massive amounts of genes. Now, when we do gene set enrichment analysis for this huge list of genes does not sort of parse out with any coherent transcriptomic signal, nothing significant. This smaller number of genes, we do get significant enrichment for functions related to protein metabolism, and especially perhaps protein provisioning for the host. But in the depleted animals, we're sort of interpreting this as like a haywire transcriptomic response that in the face of heat, without their proper symbionts, the host genome doesn't know how to respond. So we're following up with more experiments here, especially wanting to do some feeding trials where we may be able to supplement protein and maybe rescue some of these effects. But some of that is work that Samantha Fontaine is going to be leading, perhaps collaborations. So my questions in the lab are going slightly a different direction. And so I have become really interested in how relevant our environmental variables really are out in the wild. A constant question that I get are these animals ever going up to 40 degrees Celsius or even 36? So I wanted to get a handle on that. I've also wondered, how do these variables vary over space and time? So are northern populations experiencing different relationships to southern populations, or even under smaller scales? What if I had chosen the pond water from this pond versus that pond, and how would our results have differed? So that's my third point. How do pond microbiomes vary over space and time? So really trying to understand more of the system, because I think the results I just showed, I view as a very blunt manipulation, sort of colonized or depleted. And so now I really want to work to understand the complexity that we all have to work towards. So now we're really moving towards more temporal and geographic variation in aquatic microbiomes. I'm realizing that I'm sort of expanding the area of focus to the pond rather than the tadpole. And so we're really trying to understand this under a One Health context, to understand how human land use, local environment sort of cascades through these environments. And so in some ways, these questions are really just what drives the structure of pond microbiomes, but also tadpole microbiomes. And so when we started looking into the literature to get a handle on where we would go here, a lot of the work has been conducted in pristine environments, Norwegian lake systems or high mountaintop lakes that are presumably untouched by humans. And so while this information is really great for a baseline, I don't think it gives us a handle on the complexity of a human-altered world. Because instead, this is the complexity that amphibians actually inhabit. These are normal habitats, suburban environments, and so I actually think tadpoles or amphibians give us a really nice opportunity to understand both wild systems and human-altered systems. Ecologists have really overlooked urban environments or suburban environments because we think of them as altered, but they're not going anywhere. So we as scientists have to understand them. There are unique ecological and evolutionary principles happening in these systems, and so we can't continue to ignore them. So what we've been doing is some large-scale field sampling of pond environments across the US. So here we have different sites, within each of these we have about 5 to 8 different ponds at each of these. Sorry, I should have acknowledged. Emily and Jose are really leading this project. I cannot give them enough credit for how much fieldwork and logistics that they are running in managing all of this. So here's Emily here. This is actually Josh, an undergrad assistant who's helping on this project. So we've been out, well, they have been out doing all of this sampling. And so what have we found? Well, with collaboration, we have been trying to understand some of the local environments. So with Corey Richard-Zawacki, who's also at the University of Pittsburgh, her group is really focused on chytrid fungus and trying to understand the dynamics of that disease. So in some ways, they're interested in the same questions. How are environments altering disease dynamics? And so they have been monitoring pond temperatures in some of our same field sites. And so here we have temperature profiles for 2 Pennsylvania ponds at 20 centimeters depth. And so this is nice temperature data because everything we've been based off of is sort of weather data or often like temperature loggers in the air. So here we have actual water data. And what we see is that moving into the summer, we do see that the aquatic environments in these ponds do experience high temperatures. Perhaps not for 20 days at a time, sustained hot temperature, but we're trying to get a handle on what the natural variation is. And now we're really trying to understand what some of these massive upswings in temperature are, and maybe that's what tadpoles are experiencing. Another way we've sort of seen that these animals experience heat waves is just through repeated sampling of some of our sites. So here we see a pond in May that is about 2 feet deep, in June only about 1 foot deep. And then just recently, Pittsburgh was bracing for the longest heat wave in years. And so as I'm walking to work, sweating, I think, oh my gosh, we just sampled. And now we have the heat wave that we study in the lab. And so I felt very guilty going to my lab group and being like, I know you just sampled, but I think we have to sample again in the heat wave. And I'm busy. So I was really, really grateful for them to go out and do this sampling, because what we found was a real life example of our fitness effect. During this heat wave, that pond had dropped down to about 6 inches deep, and instead of the beautiful tadpoles that we usually find, they found what they described as tadpole boogers. And I think it was pretty recent, because these degrade or decompose really quickly. So I think we captured this mortality event. So this is just to say, this is a real world environment. These are real fitness effects that we're studying. So I'm excited to continue working on this system. So back to our more wide-scale sampling, because those results were in Pennsylvania. So we're starting to think about these large-scale environmental variables. So here we have latitude of our different ponds and the temperature. And so, of course, our northern ponds are warmer than our southern, or our northern ponds are cooler than our southern ponds. And when we correlate that with microbial diversity, we see a significant relationship. This is of the aquatic microbiome, so the pond water that we have collected from those environments. So we're seeing some relationship between pond temperature and available microbial diversity. Another variable that has come out as interesting is conductivity, which can be used as a proxy for salinity. We're developing more accurate measurements now, but conductivity at the time was a good start. And this is we see that our northern ponds have higher conductivity. And I think in the US, this is probably a result of massive salt use, especially in winter systems, through winter storm systems. And this is a new area of research. We've sort of overlooked salt pollution, even though it is very visibly obvious living in the north. I can't believe how people dump out salt. So this is an area that I'm really interested in following up on. So when we look at the pond microbiome, we also see a relationship here where saltier or ponds with higher conductivity seem to have more available microbial diversity. But we're very much just starting to understand the complexity of these systems. So going forward, we want to collect richer metadata from this system, collaborating with folks to get GIS data, perhaps on road density, vegetation data. And then after that, we'll really move towards variance partitioning, because a lot of our variables are correlated with one another. Northern ponds are colder and saltier, and so how do we parse those effects apart? And I guess if anyone hasn't heard of variance partitioning and you are trying to deal with multiple variables, it's very nice to, rather than to say it's this variable or this variable, you're able to say it's mostly explained by this, but it's also sort of explained by this. And I think that's the reality of the systems we're working in. We're not going to have these binary answers. And so we have to, this I think is a way towards moving through some of the complexity. And then we're also interested in going from that landscape back to the laboratory. So once we learn what there is to learn, or at least some of it, out in the wild, how can we bring that back to the lab? So, for example, maybe we could bring microbiomes from rural ponds and urban ponds into the lab and see whether those confer differences to the tadpoles. And we've started to do some of this. So graduate student Elizabeth Rudsky just started by asking whether we can inoculate tadpoles with different pond microbiomes. I think this has a lot of interest, both from an ecological perspective, if eggs are laid in this pond versus that pond. And then also thinking about, how these impart differences or differential effects on the host. So sorry, they could be laid into that pond, or we could be the ones using these different pond waters in terms of amphibian breeding and re-release. So I think this also has more of an applied use to say how we, or which pond we collect water from could have impacts on the animals that we're then releasing out into the wild. So for this experiment, we are just starting with 2 ponds. So we collected pond water from 2 different nature reserves in Pennsylvania, filtered it to remove the massive vegetation, insects, and then store this massive amount of pond water in cold rooms at our university. We did our same breeding regime in the lab, and then are raising animals in the different pond waters, but also different concentrations of pond water. So here we have we either used 100% lab water or 0% pond water, so this was our control, or we spiked in just 1% pond water, so is just a little drop enough to inoculate our tadpoles. We then used our 25% treatment, and I failed to mention that this was actually the treatment for our previous experiments. We used 25% pond water and 75% lab water because it's a lot of work to move all of this pond water around. So we also had controls within this treatment. In the last experiment, we only used autoclave treating, but we've added another treatment, which is filter sterilization, just to make sure that we're doing the best we can to control these systems. Then we have some middle pond water treatments, and then up at the high end, we were raising tadpoles in 100% lab water. And so I think this also is really important from an applied standpoint. If we think about breeding amphibians under captive conditions, we can think about what pond does this water come from and how much do we have to add. And I will say it's a lot of work to move 100% pond water. So from an applied standpoint, we don't want to do that. All right, so what have we found? We are able to successfully inoculate these tadpoles. So when we look across our control treatments, again, they have a microbiome. We're not creating germ-free animals. But these control treatments all have similar diversity to our lab water treatment. In what we are calling our microbial mass effect treatments, I'm open to discussion on that. I think it's a working term. But we do see differences that when we add more pond water to their tanks, they end up with a higher diversity of microbiomes, at least when given water from our PLE station. Because when we look at our other pond water treatment, we do not see this effect. As we add more pond water, we don't increase the diversity of these recipient animals. We can also look at this in terms of community membership and structure using some of these ordination plots. So here we have the points showing animals in lab water. All of these open symbols are the controls, and these points are the 1% treatment. So maybe they shift a little bit, but they are still very close to all of these controls. When we look at the inoculated groups, we see that they have different microbiomes from our controls, and they cluster depending on the pond water that they were housed with. And we see some confusing concentration effects. So if we give them 100% pond water, they end up with a different resulting community than if they are given 75% pond water, or 50%, or 25%. When we look at this in terms of community structure, incorporating relative abundance, we see some similar trends where in our control and low pond water treatments, they're more similar, and at high pond water treatments, we're seeing this divergence. So honestly, I haven't thought about what this means for the future of our experimental design. I'm still sitting with these data. But it does show we're able to inoculate with different microbiomes and start to think about how these different communities impart differences on the host. I'm going to flip through this pretty quick. This is similar to the previous graph, but just to show that we see differences in community structure depending on how much pond water is given to these tadpoles. And one thing I think is interesting is maybe more stochasticity in 25%, and so maybe with more pond water, we're giving a more consistent inoculation, but we need to work through that. And then finally, thinking about the effects on the host. So I'm just going to show data for body mass. So here we have our control, all of our control treatments really. No differences in body mass here. When we look at our inoculated groups, we do see a significant difference. And here, body mass was actually larger in our colonized animals. But the result I'm most interested in is this difference. These groups were both given 25% pond water, although maybe the 25% pond water isn't the right one to compare. I'm thinking this right now. But we're seeing a difference in body mass between these groups, depending on what pond water source they were given. So we're at least seeing this differential effect on the host, and so this is really giving us the beginning to move this work forward. So summary for this section, my second section is much shorter. Microbial communities contribute to thermal tolerance with implications for fitness. We're still working towards a better understanding of the mechanism. We're thinking there's something about protein provisioning that we're following up on there. But really starting to think about more variation and embracing this complexity to understand landscape and time and how these different variables impact the whole system. All right, so I see the clock. I'm going to work through this section. Just to highlight this real quick, where we're working with a conservation agency to try to understand whether we can use these host microbe interactions in order to improve animal health. And this is work with the International Crane Foundation, with Dr. Barry Hardup, who was the lead aviculturist. He's now retired, and Jess Diaz, a student in the lab. All right, and so here we're wondering, can we harness the microbiome to alleviate problems that we have with the captive rearing of these animals? And I really couch this under a larger idea of adaptive management in the built environment. As conservation issues continue to rise, we have been bringing more animals into captivity. We know that captivity alters the microbiome, and I think it's a good question, this idea of rewilding. For some captive animals, that might not be a problem. They might have the zoo microbiome that they need. But these animals, I think, are more important. These animals are going to be re-released into the wild. And so we want to make sure that they are given the proper microbial context to go out and thrive. And so we've been working with the Crane Foundation that works to breed endangered whooping cranes. This is a critically endangered species in the US. It was hunted down to 21 birds in 1941. Now there's there's about 500 individuals, but this is still very far from a sustainable population. And it's still dealing with conservation issues. In fact, the USGS breeding program was ended by the Trump administration in 2018. And so we're facing new hurdles or a slower process in breeding these birds. And so at the Crane Foundation, they raise more birds by rearing some out as being artificially reared. So you can either have them parent reared, but the cranes can only care for one chick at a time. So you can siphon off some eggs and raise them in these really fun costumes to create these artificially reared birds. So this allows the Crane Foundation to produce more birds. However, these artificially reared birds have different growth patterns. They have a higher rate of skeletal deformities, often in they have little splints that they have to put on. And they also have higher rates of respiratory illness and mortality. And with how much work and money and energy goes into creating these birds, any loss of an individual is extremely devastating. And so the Crane Foundation really works under the idea of how do we build a better bird? And so that's where we thought maybe differences in host-associated microbial communities could underlie some of these differences. And so we've wondered, can we rescue the microbiome and even perhaps ameliorate some of these health issues that we've observed? So we've really just been inventorying the communities of parent-reared and artificially reared birds over time. And then we have another group. This was a lot of work. We've done fecal transplants in whooping cranes. This was a lot of work through ICF to get approval, to get them on board. So we ended up creating a fecal slurry. Well, we collected feces from adult cranes, created a slurry, screened them for avian pathogens. So it did have to go through a freezing process to screen for pathogens. And we've been inoculating that into some birds. We only have a sample size of 5. You'll notice 2020 is missing here. This has been a complex project to run, but I think we've gotten some rewarding results. So what we see is that in our parent-reared birds, diversity decreases over time, but we see a massive difference in our artificially reared birds. Especially during this early time point, they have a much lower diversity than our parent-reared birds. But our transplant does not seem to rescue this in any way, at least in terms of alpha diversity. When we look at beta diversity, we see our parent-reared birds here, higher variation in our artificially reared birds. And in our FMT group, they fall somewhere in the middle, and they do have lower variation than our artificially reared birds, but I want to do more statistics to control for year and stuff like that. So put a little asterisk by that result. But it could be that the FMT constrains that variation, that rather than this high stochasticity that we observe in the artificially reared birds, they get a little bit of a boost in structuring their microbiome. But we've at least shown that these conservation practices have effects on the microbiome, and that we might be able to harness it to improve these conservation outcomes. So moving forward, we're going to connect all our microbiome data with health parameters. So we have all of the narrative level or bullet point vet notes from every single day, because it's been really interesting to work with this agency where the cranes are the priority. And so if a bird sneezes, they get antibiotics. Because they're working under different priorities than we are. And so it's been a really helpful learning process for me to understand how they work and then how we can conduct science under their context. And I think those types of collaborations are going to be important and necessary to address some of these problems that we're facing. And then I wonder if we over-engineered the FMT. We had to do some of that because of the protected aspect of the system. But if I could go back in time, I think we would just move poop from one enclosure to the other. Because in fact, these cranes consume the feces of their parents, which is not in the academic literature. I only know that because of animal caretakers. And so I think just moving over poop would have been a more successful strategy. So I'll leave with this image that we are drastically changing the world in many different ways. But I think we also have the technology and the power, especially through the use of microbes, in order to counteract some of these effects. So with that, I'll acknowledge a number of collaborators and funding sources that have supported this work, especially the agriculturists at ICF. So I did get to wear a little crane costume. And then I want to thank my team. These are really the folks that are doing all the work, going out in the field, sampling during a heat wave, extracting samples, things like that, and make it really enjoyable to go to work and give me the opportunity to come here and share all of this fun work with you all. So I'm hugely grateful to this team. And then I think I'm out of time. I'm happy to take questions at the break. So thank you all.
Thank you very much, Kevin. Yeah, I think we'll save the questions for the break.
Good.